I could ask the rest of the fire team to come up, and then if you guys have questions that you would like to ask the fire team, we can get started on that. And I do have one for Kevin already from online. So, yeah, so you had mentioned that you were going to make the UBE3A in factories and then get it out to the rest of the brain. Someone online said, well, then how is it going to get into the cells to actually do its job from the factories where it's being produced?
Well, I don't want to get into all the details, but it's very similar to what Dave is doing, actually. And we put these tags on the protein, and the tag will not only tell the UB3A, oh, I need to get outside the cell, but there'll be a second tag that says, oh, I should go back into another cell. So essentially, we're tricking the system using its own mechanisms in order to make the protein for us and then engineer it so that it gets taken up by other cells.
And we had another online question for Dave Segal. And the question there was, can you explain how your approach is different than the antisense approach that's being taken by ISIS? ISIS, the company, not anything else. Yeah, I think they're working on it.
I can just talk into this. Right, so ultimately there are different ways to reduce the antisense from silencing the UB3A gene. So the antisense is one method, and we're trying this transcription factor approach as a different method. The antisense method is an anti-RNA approach, in that the antisense goes in and what we believe is happening, we being the scientific community, I think, is that it's actually cutting off the end of that RNA, that antisense RNA, and not allowing it to somehow interfere with the UB3A. Ours is to try to reduce transcription of that antisense from the point where it's actually made. And there might be pros and cons of either method, but there's a little bit further along, and that's very encouraging. So I would say that ultimately it's another method to inhibit that antisense RNA and to reactivate UB3A in the brain.
Thank you, Becky. So I have a hypothetical question, which I'd like each one of you to answer if possible. So let's assume you each have a child with Angelman, and you each have $10 million in a bank, has to be earmarked for Angelman research and development. So you have a child, you have the money, where would you be spending it right now?
And I guess you're not allowed to say on your own work, or?
You certainly could. I mean,
okay.
It's open, so it's your money, it's your child.
Okay. Well, this will be fun.
Do we start?
Go, Scott.
Is this good?
Yeah.
So I think, Sharon, you asked this question, didn't you? We've already been asked this question. And this is actually where, for me at least, this is where the FLY project came from. I felt that would be, now $10 million wasn't on the table, but in terms of identifying something that, or a method or an approach to finding a new therapy, that's one that I felt that would be the best opportunity to maximize your chances to identify something. And then those that are found there then move into the other models. But that was my answer at the time. And that's really where that project came from.
I would probably give it to FAST, actually.
Of course. Your check's in the mail.
Yeah, I mean, it's a difficult question because there's a lot of horses in the race, right? So, it's funny how, at least in my experience, you're looking at one line of research, and then from that single line of research, there are several different things that come off of that. I think that this is one of the, as you well know, the kind of organic parts of science. And so it's really difficult to say, what you'd want to put all your money into. It's probably better, as was mentioned earlier, that what we're going to probably be seeing in the future is multiple therapeutics that are going to be targeted at different areas. So I think it's almost better not to put all your money onto that one single horse. That would be my answer. But fast is number one.
Yes.
I have this, Ed. I agree with Ed. I wouldn't. Sorry about that. I agree with Ed. I wouldn't put all my eggs in one basket either. But I certainly would fund my own research. I believe that gene therapy is definitely an up-and-coming tool that could be very useful. I mean, not in the immediate future, but maybe 10, 15 years from now, once we get things perfected. But I think that other avenues of research is also important, because you may have to target more than one thing in order to get a global effect. You might not get that with one treatment.
So, without trying to sound like I'm saying the same thing, I would think that the question kind of is asking, if we weren't resource limited, is there something else that we would be doing? And it's hard for me to imagine, that what else we could be doing at this point as a community. Of course, having more money always makes it go faster, I suppose. But, we have a lot of very interesting basic, kind of preclinical research going on here, investigating things in a couple of ways. Even the last question that was asked, what's the difference between the antisense therapy and what I'm trying to do? I mean, you see multiple different ways being applied here, so that in the end, that it's not just my way or, Kevin's way, or one way that we're just going to put a big bet on that and say that's probably the one that's going to work. Let's try that. I mean, at the end, you're going to know that we're having the best therapies, or therapies that can apply to as many people as it can, because we have many different kinds of efforts going on. So I guess something that might be nice to see is if we had even more people involved in doing the research, if we had even more companies involved. But, the number of researchers that are working on this, sitting here and then also around the world is, I think, very exciting now. With all the advances that have just been made in the past couple of years. And seeing all these companies at this meeting now is also really very exciting. I mean, just in the short time that I've been involved here, I think this is a tremendous development, to have people that can really move things to market, coming to this and starting to make a difference. So, could we apply that money and make things maybe go faster, maybe get more people involved or something like that? But I think that already we're seeing a lot of different approaches being tried and explored, a lot of different ideas, and then a lot of dedicated people, especially the families, right, that are supporting all of this research, because that's where a lot of the money has been coming from now, and a lot of the energy to do this work as well. And so, everybody has a role to play in this. Like I said in the past, you feed off of us, we feed off of you. So I don't know what I would do with the $10 million, except maybe try to give it out and see that we can do more of what we're doing.
Yeah, I agree. Diversifying the investment, trying to get more people involved, but that's basically what's already been said.
So can I add one thing to that? I think one thing that I wish we could do, I don't know that there's a way to do it because it's not very directed, is I wish we had enough money and enough resources to start going through all kinds of either FDA-approved drugs or things like OV101 that have been through a certain amount of work and maybe could go further. I think right now we're having to do a little bit of picking and choosing as to which of those compounds we look at, and sometimes we get lucky and sometimes we don't. But it would be great if we could do that a little bit more unbiasedly and just try everything. It's not realistic, but that would be one thing I think we're not really doing. Allison. Tina?
Hi, my name is Tina Thompson. I actually have a question about maybe more directed toward the Ovid panel this morning. Is that kosher to ask now? So I heard two things during the presentation this morning. One was a reference to animal models and how OV101 had worked on the animal models. And then I'm not sure if I picked up right, but at some point there was a dosage question and there was a reference to using 1/10 of the dosage of Alzheimer's patients. So I'm not aware of either OV101 being used either in animals or on Alzheimer's patients. So I wanted to just hear a little bit more about what had been done there and what the results were.
Okay. Yeah, so sorry to the panel. Yes, I was happy to take that question. When the drug was first discovered, it looked like a GABA molecule, and it was a time in the history of neuroscience where we didn't have the full understanding we have today, approximately 20 years ago. And at the time, we did know that certain diseases of the brain had deficiency of GABA neurons, Huntington's being one, Alzheimer's being another, where there's a loss of GABA neurons in certain parts of the brain. And so the simple, naive idea at that time had been, this drug looks like a GABA molecule, let's just give it to these patients and hope, and use as big a dose as we can. So they started with doses 10, 20 milligrams 4 times a day, escalated up to 120, 160 milligrams a day. And the patients were still walking around for the most part and still okay. So the drug was safe, but they did very short studies. These were open label. They're in very severely affected individuals, and so they really weren't pursued. And in retrospect, they weren't well-designed studies. It was used in many other indications. It was used in pain, was used in one trial in epilepsy. It is an analgesic. It does have anti-epileptic activity, but none of those really stuck, and it really didn't really pursue from a commercial point of view, from a drug development point of view, none of those indications really looked ideal. And it really wasn't till this concept that it acts on the selective extrasynaptic pathway, which was a much more recent finding, that all of a sudden there was a sort of crystallization of two events. One, understanding what we call the molecular pharmacology of the drug, so we knew exactly where in the brain the drug was working, not just like a GABA molecule. And secondly, an increased understanding of the biology of this disease and biology of Angelman, and those two bits coming together. And really, the reality of the drug really wasn't used in an animal model specifically as a therapeutic. It was really to prove that tonic inhibition was important, and restoring tonic inhibition could improve some of the function of that animal. So really, it was that basis, and seeing that biology and seeing parallel studies occurring in many organ systems, both flies. There's some very recent papers now showing that if you can actually use Drosophila, not Angelman Drosophila with other cognitive deficits, you give OV101, you improve the cognitive phenotype. If you give the drug to Fragile X mice, you also improve them. If you give it to Rett syndrome mice, you also improve Rett syndrome. So all this data sort of accumulated to a point that we felt sufficiently comfortable with the drug, with the pathway, that we felt it was appropriate for us to move towards the clinic. So I hope that sort of helps a little bit.
Yeah, thank you.
Okay, Allison.
I'm going to first thank Dr. During and Ovid for donating $10 million to the fire team at FAST, and we'll all be proud to take that. But along the same lines, actually, my question does go back to that. Knowing, and I asked Scott this question yesterday, but I actually want to ask all of you the same question that I asked Scott yesterday. The title of our organization is FAST, and obviously things are moving very quickly with that title, and it's, for us parents with children with Angelman syndrome, it's not fast enough. And even though it's quicker than you've ever seen before, what is it that could be done, or what is the rate limiting step to make this faster? If that's 3 months faster, 6 months faster, 2 years faster, is it money? Is it time? Is it personnel? Is it the science? Is the rate limiting step things that cannot be moved any faster, or are there things we can do? Because I think financially, the iceberg hasn't been touched with what we can do and what we can raise. And I think if we knew what we needed to make this move faster, I think we could. And I think we need to know what that is.
Yeah, as far as rate limiting steps go, taking something to clinic, I think that Dr. During addressed that earlier today. I mean, you can only go so fast through making sure that something is safe, making sure that you get approval through the FDA, going through, all of those steps. What can you do to make things go quicker? I think for now, it's some of what Jody talked about, the registry, making sure that everybody is registered so that when the clinical trials come on board, it's going to be easier to find those patients. Coming out with good outcome measures so that it's not, you don't have to replicate a clinical trial over because you didn't do the right outcomes. I think that being proactive in that way is going to save us time in the future as well. But I think we're at a point now where things are changed. I mean, you can see there's companies up here that are talking about going forward and how fast they want to go, but there are limiting steps. Sometimes it's not the science, it's just how those things work to get to clinical trial. So, but I think we can be proactive now to save time in the future.
But what about like with the fire team? What does the fire team need to be moving some of this stuff from just getting started or just getting good results to being able to pass it off to an Ovid or an Agilis?
Yeah, well, I think that Scott and I are both doing drug discovery. I mean, if you had a whole team of people doing drug discovery, it would go much quicker. There's subtleties here too. So one of the things that Scott and I talk about when you're doing drug discovery, it really is kind of a fishing expedition. You're looking at thousands of different drugs. Some of you don't even know what they are. Some of them are soluble, some of them are not. Some of them are yellow and green, and you don't really even know what you're putting them on cells, and you're hoping to see a result. You're trying to do this as quickly as possible, but this isn't hypothesis-driven science. So in order to get that science to get moved forward, it's difficult to give somebody with a lot of training that kind of a task, because this does not promote their career. This isn't something that they can publish. It is just grinding work that takes a lot of time and energy and effort. And a lot of times you have to do that with volunteers or technicians, somebody who's not at that higher level. So, that's what throws that into a much slower process. And
Well,
I think
but
Scott would agree with this.
But we also have this whole idea in science with the brain drain, right? There are plenty of people who get through with their postdocs and there are no jobs for them, right? I mean, we know this. So if you had the money to pay somebody a decent salary to come in your lab and be an expert technician to screen drugs.
Yes, most certainly. Yeah.
Okay, well then let's do that.
You'd have to do that.
That's what we need.
More money you could definitely throw more money at that and more personnel at doing something like the drug discovery work.
Yeah, so one of the things Ed's talking about, for those of you who aren't thinking about academic research, is that we get graduate students and postdocs into our labs, and it's being an apprentice, like a journeyman. And so we give them projects for them to work on and work their skills out and use their own intellect to move them forward. And then that's supposed to help them get their own job. And so you can't give them something like, go look at 10 things and see if something works, because if it doesn't work, you've kind of killed their career prospects. So, in that way, probably money for people who are experienced and want a job but aren't going to be hampered by this kind of project would be helpful.
And it's also not something that would be funded by NIH because it's not
Correct.
hypothesis driven. They don't want to fund a fishing expedition, look at 10 or 20 or 50 things and see if something works. They just won't fund that.
So
right or wrong, it doesn't happen.
Yeah,
Yeah,
that's a mistake.
That's exactly the
but-
second thing I was going to say, is that this is where foundation support is just invaluable, and you can take that money and do things that are high risk, but potentially have a high payoff. And so the more money that you have to do those kinds of experimentation and research, the more likely you are to get that one, big, huge hit. When you're writing grants to the NIH, it has to be bulletproof, and funding right now is very, very difficult. So it almost has to do, you almost have to propose things that are going to work, and they know are going to work. This isn't how science should be done. We should be taking bigger chances. The foundation money allows us to do that, so we can come up with really innovative ideas that are potentially high risk, but could have a huge payoff. And you never know what that next identification of a therapeutic may be in order to move that forward and attract another pharmaceutical company or another biotech company. You know, the work that Jen Daly did was not on an NIH grant. That was actually a foundation grant that we got. And that science really kind of drove things forward for Agilis to come and talk to me and get interested in the research. So that kind of money, it's hard to say, well, this is exactly where I would put it, but those kind of ideas that get funded really can drive the science quick.
I think what slows down a lot of, especially in the research that I do, is the animals. I mean, they are animals. You have to breed them, you have to grow them, you have to wait for them to mature in order to use them. And you also have to screen them and make sure that they're the correct animal. I mean, we have to make sure they have the mutation. We don't want to use animals that don't have the mutation. So there's a lot of time and effort and money that goes into maintaining the animals and making sure they're healthy, making sure they're growing, making sure we're getting more of them. And that's also a big expense that we have to also consider. And it takes a lot of time. Because even the studies when I do the injection, I don't do an injection and then the next day I analyze the mice. I have to wait for them to, produce the protein to see if there's an effect. And sometimes some of the studies we do, we wait months and months, 6 months, waiting for that effect to come through. And so each day we have those mice, that's more money that it costs for us to maintain them within the facility.
Dave or Ann, either of you want to add to this, or should we move on to the next question?
Just to say that I think that, yeah, something that you could do with the money is just involve more people. So more people in the lab work, more people coming up with ideas, more people. Yeah, you could use the money for more people.
Yeah, it takes an army to do things the correct way, and you don't want bad data. You want it done right. Behavioral studies like what Ed and my lab do, you have a large sample size, and I mean that's just what's required, and it takes a whole lot of time.
So personnel, and so this is why we were saying this morning, we still need you to fundraise for FAST. We still need money. There's places for that money to go that's still going to be important for us.
I want to know as a parent how we can help, and if you guys, gather what kind of information, testing that you can help quantitate. Eventually, what kind of testing would you like to have our children for baseline testing? So if we go in yearly, we have the new AS clinic in Mayo. We'd love to have that voice output or the gait monitoring data that you were explaining earlier. What kind of things would you like for us to start, that we can maybe start collecting baseline data?
Yeah, so we'd like to have a symposia. Jody is kind of spearheading this whole thing with Agilis, and what I think we're trying to get is a consensus from the clinicians, from other people, to say these are the things that we can start looking at and establishing baselines now for the next upcoming clinical trial, and that we can use. And it's going to be a whole kind of menu of different things that we want to establish, be able to look at, to refine, get people's input on how to do it and how to do it correctly. And then those things can be chosen for a particular clinical trial depending on what they're looking at or depending on what's being targeted. I think what's important now is really the first thing is the registry. So the registry is going to be huge, and it's not just a U.S. or an Australian thing. The registry is going to be something that is going to be, people from all over the world are going to be able to register, have all that information put in so that when clinical trials come up and they go outside of the U.S. boundaries, people know what's going on and can be part of that clinical trial. So I think that right now we will be asking the community, this is what we need you to do. We need you to come in and help us establish those baselines. But for now, we just really have to come up with some kind of a consensus on what those baselines are. Okay. We're all of the idea that new outcome measures are needed. Yeah. So now we need to really work on what they're going to be, what they're going to look like, and what can be FDA approved for clinical trial later on.
Okay, thank you.
So just to follow up on that, as a clinical provider, I fight daily. Even here at this meeting, I'm fighting with insurance companies for reimbursement, for approving, pre-authorizing a test. And I think the reality of this is that it's going to take time to do each one of these studies, to do the gait analysis, and the hospital and our academic institutions won't let us do anything for free. If it's research, there has to be a research dollar that covers our time, that covers technician times, that buys the space to do the test. So I think with our $10 million, part of what would need to happen is to sponsor once a group has met and there's a consensus on what's needed in terms of additional phenotypic characterization, then and even genotypic characterization, then we're going to have it funded. There may be some cases where insurance will cover it, but they're only going to cover it if, as the provider, I can say I need this test because I'm going to do X, Y, and Z with the results. And they get stricter and stricter with what they will authorize and cover. So, and we can't have our community of families stuck with huge bills for the characterization.
Is the pig ready now? Oink, oink.
It's like, there's so many jokes that I can make of it. No, we're in the first process of it. So, as I mentioned, they have some data suggesting that, it's been deleted. The technique that was used, very similar to what Dr. Segal described. And so from there, then the animals are cloned, and we just wait for that to happen. So we hope, there are no guarantees on any of this. He is one of the best, if not the best, in the country or the world. But that sometime in the spring, we'll start seeing pigs.
Do you think
a spring pig?
Mm-hmm. Would that be before it's even needed? It kind of sounds like it is.
That would be good.
Yeah.
Yeah, you want it before you actually have to have it.
Yeah. Anyway, I have a question for the last person. I'm sorry, I don't remember your name. But
Yes.
If the patient has an issue with their brain, like an intraventricular hemorrhage, or if they have some other brain malformation, would a medicine delivery that goes into the brain, would that not work on certain people maybe? I don't know if that makes sense, but.
Yeah, you're saying if they have some sort of defect, maybe in the ventricular system or something like that.
Yeah.
That is difficult to say. It depends. I would assume if it's just a structural thing, it may not be a problem, because you would still have the cells lining the ventricular system, and those are the cells we're interested in affecting. So they still may be functional, and we still may be able to produce the protein, but without knowing exactly what the problem is, it might be difficult to say. But that's why we're trying different approaches. And so maybe that particular approach may not be very useful for that patient. We may be able to try one of these other approaches in order to achieve something similar. And then that way we can circumvent that problem.
Thank you.
Matthew?
Sorry, well, I don't want to, this is really the forum more for the family, so I don't want to take up too much time. But I guess just to go back to your point, which I think, when I first moved into the Angelman area, I could say, and look online and go to PubMed and look, there's a lot of great science in terms of animals on UB3A, but there's a paucity of good clinical studies. And I'm not just talking about intervention, but we really don't have a good understanding of clinic. We know a lot more about the mice, far more about the mice than we do about the patients. And I guess what I'd like to ask again, each of you, is to think about, again, what would you really want to know? What is missing from the clinical data out there that would really help shape and advance your research? Because I think there's some huge gaps out there. I'd really like to know your thoughts in terms of what you think is missing, not just in terms of patient registries and what you're doing, but what would you really like to understand about the disease and about the patients as opposed to a pig or a mouse or a rat at this stage?
I think one of the for me, it's why there's such a variation in Angelman syndrome if it is monogenic. If it's just one gene, why are all these kids so different? So that understanding of gene-gene interaction, what makes one child be able to learn to walk when they're 2 and another child not until they're 11. There's such a wide variation in in the symptoms of Angelman syndrome, there's got to be other interactions in there. I find that, you know, it's got to be very complex, but a very fascinating area that really has not been touched.
Yeah, we talk about this a lot online, right? There's 20,000 other genes that go into making up who you are, and they have to be influencing this on some way, just the way that Dr. Weaver was saying. So,
I think for me, there's two questions that I'd like answered in terms of like a gene therapy approach. The first, I'm just making sure this was on. The first question is, do we need to target the whole brain? Or is there particular structures in the brain that are most critical? And if we can target those only, would that be enough to affect the quality of life of the patient? And the second question is, do we need the entire protein? So UB3A gene is actually quite big, and the protein is a fairly large protein, but it has different domains. And the question is, do we need the entire protein? Because sometimes if we can make something smaller, it might be much easier in order to generate the therapeutic that I'd like. So if I can reduce the size of the protein that I'm making, it may be much easier for me to get the therapeutic benefit that I need. But right now we just don't know, do we need the entire protein, or is it just some of these domains within the protein that is critical?
Well, on a research level, I would like to know how loss of a ubiquitin ligase leads to the symptoms that we see in kids. I think we have a description of what Angelman syndrome kids are like, and we know this gene defect and the gene that's involved, but actually going from that gene defect to the things that we actually see is not a clear pathway to me. We know that UB3A has a lot of targets. We don't know what all of them are. We don't know what are the ones that are most critical for causing the disease. And so I'm often struck by the fact that there seems to be this kind of phenomenological level, kind of like Dr. Anderson here, you know, measuring the EEGs and things. And this kind of genetic level where we know what the defect is. And then there's a whole region in between where we don't really have a good connection between how the loss of this ubiquitin ligase is causing the things that Dr. Anderson sees. So if we knew all the parts of that pathway completely, that may provide other therapeutic targets, or it might provide a much richer understanding to start, you know, thinking about to develop therapeutics in. But that's on the research level. On the clinical level, I imagine the kinds of things that we would need are the kinds of things that were brought up this morning. We need good outcome markers. Like all the studies that I show where you could see UBE3A coming back on in the brain. Obviously, that's not something that you can do in a person. You can't see the UBE3A come back on in a person because you're not going to take the brain tissue out. So, you know, what are those clinical markers that we need? What are the biomarkers that could tell us how well the therapy is doing, or if someone is going to be more severe or not? Those would be nice things to have.
I agree. I mean, along the same lines as what others have discussed. I mean, it's my big question is related to epilepsy. Why do some children have severe epilepsy and they go into the subclinical or subtle clinical status epilepticus? And what's different about those AS children or adults compared to the ones that maybe have a few seizures or pretty well-controlled seizures, or that have no known seizures at all? You know, is it related to their mutation? I think we have enough data, even though it's not comprehensive, to suggest that, well, those that have the GABA receptor included in their mutation, yes, those children often have, or AS individuals often have worse epilepsy, but we also know just the UB3A mutation itself can be associated with severe epilepsy. So we really don't understand the basis of that part of their phenotype. And if you're continuously seizing, that that likely affects your cognitive phenotype, and you as parents, I think, know that. You probably know when they start having subclinical seizures because you lose the interaction with your child. So if we understood that, I think we could direct therapies better and maybe come up with a cure.
Okay, thanks. I actually want to ask a question about the outcomes, and forgive me if I'm misunderstanding. So when you're submitting for approval, you want to submit certain outcomes to the FDA so that we can approve the trial of the drug. Is that correct? Yeah.
So yes and no. So you can, for your primary outcome, you have to get approval for that. So you can't just come up with any outcome that you want. For secondary outcomes, you can change that. So you can look at a lot of different things. I think that the key here is, I think there's a lot of confusion on, even from us and clinicians, on what you need to do and how you need to get that approved so that you can have that as a primary outcome measure. So, there are defined primary outcome measures that are FDA approved, like the Bailey's, like the Vineland, the same ones that I used in the minocycline clinical trial. I think the key here is that those are not specific to Angelman syndrome, and the NIH has even come out with potential grants that you can apply for to come up with new outcome measures specific to different cognitive disorders, Angelman being one of them. So I think they understand the need, and they're trying to actually come up with ways to standardize outcome measures so that clinical trials can be, assessed, across the board. It can be compared a little bit easier. But I think the problem that I found in the clinical trial that I was involved in is that those outcome measures are not geared and not specific to Angelman syndrome, and I think that's what we need to do. So how we do that, how the best way to do that, having FDA input into this once we come up with these ideas, I think is critical. But from what I showed you earlier was strictly to show that there are ways that you can look at different outcomes, and you can do that in a way that's not subjective. And I think that's really what's needed.
Sorry, this is probably more a comment than a question, because isn't it that we're trying to get a therapeutic out there specifically for Angelman syndrome that is going to improve the motor skills, the fine motor skills, the hopefully speech, that kind of thing, no epilepsy, or reduce the epilepsy levels. So how would you, can't even format the question, how would you put that to the FDA and say, this is the outcome we want, but this is only, this is across the board? Is that making sense? No?
Matt, do you want to answer that?
Sorry.
The problem, and Ed and I have discussed, and I think Ed's done really some pioneering work that he presented some of the data here, which is really a great step forward. There's multiple issues here, because remember, if you're looking at within the Angelman community, there are patients who can't walk, there are patients in wheelchairs, there are patients who need sidearm assistance, and so you've got this incredible variability from the first place. So if you say, well, let's look at the gait, the other point is that only certain subjects will be able to go on the gait. There's lots of endpoints that have been used to approve other drugs, but they're not really relevant to this population, as Ed has stressed. And for us, the issue is really one of, you also want to say, what is the change in that endpoint? You can measure someone's along the gait, but does that really reflect clinical significance at home in terms of lack of falls, in terms of independence, in terms of mobility? So whatever endpoint you choose has to be relevant to the real world. Is it really something that clinically stands and is meaningful? So these are really difficult questions. And I can tell you that one of the most advanced companies in the rare orphan space, a company called Ultragenics, has developed a whole team, and that team is just entitled endpoints. That's all they do, because that is how important this question is. So I take my hat off to Agilis to work on this. We clearly at Ovid are taking this issue very, very seriously. And as Jeremy said, this is something we do in partnership with the FDA. Ultimately, the key is, if you believe your drug works and if you can show it's safe and effective, many of these questions won't get answered until much later in development. You really want to design a trial that you hit certain endpoints. If you believe the drug is good, then move that forward. We're taking this extremely carefully. It's one of the things we're spending more time on than anything else. And we think we have a handle of how to move forward. But again, it's early. We need some more refining of that. And we're doing this in partnership with FDA. I don't know if you want to discuss.
Your question is absolutely pertinent. For example, in a cancer trial, there are really two measures. Question number one, does the tumor shrink? Question number two, does the patient live longer? That's kind of it. In somebody who has, and that's simple, and there's a huge long track record of that. In congestive heart failure, which is just as fatal as cancer, you have a walking test. You have certain biometrics that you can actually easily measure. What is the oxygen in the blood of the patient? You have clear measures that have been around for a long time. Here, just as Matt says and as Ed has said, we are actually broaching a new field. You have patients, as I understand it, as old as 70 years old, and as young as we heard this morning, months old, days old even. Each one showing different types of symptom. So somewhere in that, and no track record of any substantive major clinical trial through the FDA. Very important to understand that. So we have to educate the FDA. Very important to educate them. Very important to ourselves understand what it is exactly that is important and for which age group and how we're going to approach that. And then as we do that, to then think about real metrics. And we heard a lot about metrics, which I really loved that discussion. I thought it was a great discussion. What do you measure? What do you actually measure? When a child can't get out of a wheelchair, well, you can't measure motility. This is really a tough one. So I think as we look at this, the work that Agilis and others are now doing, and the NIH and ourselves, hopefully we will craft the right approach for the beginning. I can assure you that as the therapies that you've heard about here evolve, and they're very exciting to me, incredibly exciting to hear this actually, you will have to start developing, or we will, not you, we, sorry, get this right. And Matthew, you're giving me $10 million? Tomorrow, thank you, because I know what I want to do. The question is, how will we together craft the right endpoints for the right age and for the right therapy, and to make sure that they don't negate other therapies. Very important that, because once the FDA gets into its head that there's only one measure. It's very simple for them. So we're asking them to be quite sophisticated, and I think they'll be a very good partner, actually.
Thank you. Thank you.
Yeah, you also want to keep in mind that, we talk about the FDA as this big barrier to some of the stuff that we're doing, but they're ultimately tasked with making sure that nothing dangerous happens, especially to people in clinical trials, especially to children. So, it's good to partner with them, and they will hold you to a very high standard, and I think every parent and grandparent and caregiver in this room and sibling wants a very high standard.
Okay, I actually have two questions. My first is, we had a question earlier about what still needs to be done, because as parents, we want things to be done as quick as possible to fix our children, or not even really fix them. They're perfect how they are. But how much time do you think that doing collaborative research has saved our community from not having an answer? And how much time do you propose it will take? Sorry, writing a dissertation on this title.
These are great questions.
The second question is difficult. I mean, how long it'll take, you always think it's going to happen much quicker than what it does. Science is a really, really slow process. It really is. I gave a talk one time about a science experiment of dripping tar that's in a museum in England that's been going on for 100 and some years. You know which one I'm talking about. So, science it's not quite that slow, but it's a slow process. But the collaborative nature, I mean, what you're looking at up here, this whole thing with the AAV and the virus, that never would have happened if Kevin wasn't there to help me. He's the AAV guy. You know, I've got an idea. He has the expertise. This is where the collaboration will, comes together and really moves forward. We wouldn't dream about doing another large animal. I don't have that expertise. I don't have the expertise to make a rat that I can do some of the more complex experiments that I have in mind. Dr. Segal does. One of the things that we were looking at, and I'll tell you, the disruptive nutrition and the ketone ester, that was a random thing. Dom D'Agostino was actually sitting on my student's dissertation committee and said, "Oh, you ought to try this. This is, this new material that we have." We saw the potential in doing that, but I have no expertise in seizure whatsoever. And does. So, that kind of thing drives the science much faster than me learning all those techniques, understanding that type of a field and doing it all by myself. I mean, nobody's an island, and those kind of collaborations moves the science much quicker. It's hard to quantitate and say, yeah, it's increased the field so much, but I think the exponential increase in publications and how fast the science is going in the different directions is a direct result of the collaborations. And that's hard to kind of put your thumb on and say, this is how much time we've saved. But if you think about 30 years ago or 50 years ago, when not even that long, before the internet, when the way that information, scientific information was disseminated was through a journal that you had to go to the library and go, let's see what, Journal of Neurochemistry has this month, and you'd flip through and go, oh God, look what they're done. That research was done 2 years ago. Now with conferences, the internet, in PubMed, that information gets disseminated very quickly, and it can influence your research quite a bit.
Can I just say, the old adage, 2 heads are better than 1. And so, when you have 5 heads, it's much better as well. And because sometimes when you're looking at a project, you're seeing it from this perspective. But then when you collaborate with someone, they see it from this perspective. And they see something that you don't see, and then they can find things that you're missing that you may be doing wrong. The other advantage of collaborating, in my opinion, is you hear about negative data. So when you're a researcher, you never publish anything that doesn't work. So I'm sure there has been experiments that have been repeated many times in many labs just because no one reports the negative data. But at least if we're talking with each other, we're not going to repeat the same experiments that may fail. And so we can save time by not repeating things and then working on things that are working.
Can I just share a quick story? When my daughter was diagnosed, she was about 10 months, 11 months old. And I talked to Art Bodette like 3 days later and said, I thought he was going to tell me that, it's too bad, it's a horrible thing, and here's the science. And what he said to me was, do you have her in any of the natural history studies? Have you thought about doing the betaine folate trials? We will have clear roads to therapeutics in 10 years. And I wasn't expecting to hear that because that's not what I was seeing on the internet. My daughter just turned 10. So I think he was right about that. There are people in this room who have been diagnosed more recently, and they are being told by people in sort of a general way, we think we'll have therapeutics for your kids in 5 years or even 2 years. Things are changing, and they're changing pretty rapidly.
I found that in my own research. But my second question actually is more directed to Dr. Anderson, and may not actually fit right here, but it's the only time I have to ask it. So tell me how an EEG would change over a period of time. Like, it was a typical EEG for a kid with Angelman syndrome to it's perfectly normal with a different doctor and a different EEG.
It's hard to say without having the same doctor reading it.
Right.
People have different skill levels. It happens within our group itself. The more experienced EEGers have seen a lot more children with Angelman and can spot an Angelman, an EEG consistent with Angelman syndrome very quickly. The variations, and our less experienced readers are getting educated on it because we do see a lot of EEGs from children with Angelman. So I would pull both of them and have one person read them.
That'd be great, but neither one of them are here anymore. So it's just a question because the newest, we don't even have a neurologist where we live anymore. So we have to travel. And
Where do you live?
Central Illinois.
So
Talk to Paula.
I mean, we also know, that slow spike and wave that's sort of more frontally, that can suppress. It could suppress with medication, or, it could be much more evident if your child just got vaccinated or has a cold and is febrile. And then when they're feeling better and they're very alert and not having seizures, the EEG might look much more, normal.
Okay, thank
So,
you.
I mean, we know there's a lot of variability from EEG to it's
Right.
sampling error. You're not doing it continuous on your child like we are with the animals.
Great, thank you so much.
Okay, so in the interest of time, these are going to be our last questions. Anna?
Yes. Gene therapy still sounds like science fiction to me, and I know there's a lot of concerns about genetically modified food. What happens in terms of measuring side effects for gene therapy? Is there like a lot of science behind this already, or is this a new area?
It would depend on the gene therapy approach. I mean, there's with AAV, there's hundreds of clinical trials right now, I think. It would depend on what the clinical trial was for and what sort of side effects you'd be looking for. I think one of the things that we would have to look for is, maybe immune responses, inflammation in the brain, things like that. But you would have to have some sort of outcome measures again that were just measuring, neurological function. You might be looking at, EEGs or something like that to see if there's any changes in that sort of thing, so you're not generating a problem. And one of the problems might be if we make too much of the protein, we don't know what effect that would have. But we would think it would have an adverse neurological effect, so we would be looking for something like that.
You mentioned there's hundreds of clinical trials. Is there anything in market which is a virally...
That is probably better to answer that question than me. Matt's been in this field for like decades.
The only approved gene therapy in the Western world is an AAV for a sort of a cholesterol metabolism, a rare disease, a lipoprotein lipase type AAV for Glybera, it's called, and that's a company called Unicure. So there's nothing ever really got through the approval process here in the US. We have we've run trials in Parkinson's disease with a lot of work, and there's some overall the safety record of AAV. We used to always talk, and I've worked on AAV now for over 20 years, and we used to always go up and say in front of the stage, well, gene therapy hasn't worked yet, but we haven't killed anyone. And in fact, that for a long time was true. Then there was one case at University of Pennsylvania where a child had adenovirus and had an overwhelming immune response and a total sort of organ failure, and that was back in now 99. Since that time, few cases using retroviruses where it's got into the bloodstream, and we heard today a lot of discussion about the question of integration versus non-integration because of the vectors that Kevin's using and Ed are using and non-integrating. And I don't know if any of you in the audience, everyone knows what that means, but the bottom line is that because the viral DNA doesn't get inserted into your own chromosomes, it stands apart from that, it's not interfering and not taking any change, and that was a huge safety component to that. In some diseases we have to use, because if you have a dividing cell population, that doesn't work, that DNA gets spit out, so you have to have a gene or a vector that gets integrated. But in the context of the brain, I think, again, as Kevin had emphasized, in primates now we have data out 8 years. We treated patients back in 2000 in the brain, and those patients are still alive. So we have enormous safety data on gene therapy lasting, as well as not having any major adverse events. So I think the gene therapy approach can be very, very safe compared to other strategies. The biggest problem, again, as Kevin emphasized, is just typically getting into the brain and having to do stereotactic surgery, as an example, is one of the biggest risks associated with that. But as he said, he's making a lot of advances in terms of ventricular and CSF administration. I think it's really exciting stuff.
And I think if you, even though there's not really clinical trials that are finished, if you look at the number of clinical trials, In the past few years, there's a lot more that are making it onto like phase 3. So the number that are getting further in the process is actually increasing. So I think, in 5-10 years, you will definitely start to see some of these that will probably be used as a gene therapy for certain diseases. I know one of them would be there's a retinopathy that's actually looking quite promising, and there's a gene, anti-alpha chymotrypsin, which has been shown good results as well.
Thank you.
Anna?
My question is actually for Dr. Weber. Are there any updates available for the future of minocycline?
Short answer, no. We're actually doing a little bit more work on the minocycline, trying to identify what the potential molecular changes are. It's a little bit putting the cart in front of the horse now that the clinical trial results have been published, but we did have some interesting preliminary results on minocycline changes in the AS mouse brain. So we're continuing to work on that. So probably coming up with a molecular target may spur some more interest in minocycline, but as far as my research program goes, that'll probably be it.
Thank you.
Jody?
Okay, real quick. Thank you all. Those were great presentations on a summary of your research, and it was nice to sit back and just listen to you all talk about it one by one. I think that for the FAST board and the funders that have raised the money to that, it's great outcomes, and it's nice to hear that you're collaborating, but you're all kind of coming at it differently, which I think will ultimately advance the science more. My question specifically for Dr. Anderson and then for the rest of the panel, biomarkers would be great to have specific markers, and thinking of the EEG, and I heard you answer her question before, is it in the way you collect the EEG that you could interpret it differently, or is it in the interpretation? And then could we just get a bunch of EEGs from all over the place, put them together, and have an expert panel read them? And might that be a way as a marker, or is there a way to somehow computerize it and have a computer read them?
I think getting raw data, and most centers, I think big centers, save the raw data, so it's in a specific file. And then that is fed into a computer program, a MATLAB program, and you need to understand what you're doing and what sections you're taking out to analyze. But I think it would be really nice to have that as part of potentially what the group that does this consensus of what we need data we need to compile, and that could potentially be one. It's time consuming though, and it requires some expertise, but it seems like at least what we're seeing with the animals, and especially if we screen additional agents and therapies and see that if we rescue behavior, we also rescue that spectral frequency change, then that could potentially be a really nice biomarker that's non-invasive. And almost all of these children and adults too have had an EEG. So as long as you can get the raw data, it's an EDF.EDF file, and you plug it into the program. I did have, there was one online comment related sort of to epilepsy at least, and I wanted, just for the online person that made this comment, they wanted to make sure that the group, especially families with children with epilepsy, were mindful of anti-epileptic medications that are contraindicated for children with and adults with Angelman syndrome and epilepsy. And I guess it's not, we don't think of them clinically as an absolute contraindication and not an allergy really, but there is a body of data that suggests some medications can cause the epilepsy to worsen and seizures to worsen, and vigabatrin is one, or Segal. Additionally, drugs that target focal epilepsies like trileptal or Tegretol. So those are, there is a list of them, but basically I guess if you think of the epilepsy with that frontal spike and wave, it's bilateral, and myoclonic seizures and the atypical absence, those are more generalized type epilepsies, although you could see them with a focal epilepsy. But then we know generalized epilepsies can worsen when you use a therapy that really targets a focal epilepsy. So it's important to be mindful of that, and talk to your neurologist about it or epileptologist about it if they don't seem to be recognizing that.
One quick point on that before Chick gives us our last question. If you have a neurologist that you feel like is incredibly open and willing to talk to other neurologists and doesn't seem to have a big ego problem, let me know, because I would really like to put together a email listserv with, say, Dr. Tebert and some of the other really fantastic neurologists who will actually consult with each other, so that other neurologists, when they get their first AS patient, could go to that listserv and ask for advice. Because, I mean, we all know on Facebook the parents are pretty good at telling you what drugs work and what don't and what's contraindicated, but nobody wants to hear a Facebook parent tell a neurologist with lots of education what they should be doing. But if we could get our neurologists all together with Dr. Anderson and some of the other neurologists where they could ask questions in a very secure MD area, I think we could help a lot of kids. So if you have a neurologist that will do that, please let me know and get that name to me.
And I would emphasize, you know your child better than any neurologist knows your child. And if you don't think that your child is performing the way they should, I would press ahead, and it may mean you need a second opinion, and press for that. You have the right to that, and a physician should never be offended because you want a second opinion if you're not getting the response you want. It's your child, and it's your right to that. So I always say, I tell my patients, if your doctor has a problem with that, then something's wrong with that physician. That's not an appropriate response to a request for a second opinion.
Okay, last question, Chick.
Thank you. So this was a question about, I guess, gene therapy, viral vectors, or maybe even the biologicals that Dr. Segal mentioned. And just a question further about the blood-brain barrier. It sounded like, Dr. Segal, unless I misunderstood, is that you were almost a little surprised about the uptake of the drug you had injected throughout the CNS, how it had crossed the blood-brain barrier. And just to follow up on that and some of the viral vector things and the concerns about intrathecal injections, there was some news in the mainstream media this year about some techniques using like high-intensity focused ultrasound and things to disrupt temporarily and reversibly the blood-brain barrier. So you could inject drugs that would pass the blood-brain barrier and then it would repair itself. And I suspect people in this room are familiar with this. This was mainstream media stuff, not medical science journals. So I would like to know, you all in the field, is this done in one place that hasn't been repeated? Is this the real deal? Is this going to happen in the next couple of years? Is this going to drastically change the approach to a lot of these things? If you could just comment on that.
You want me to go? I know which study you're talking about, and I saw the mainstream media. To my knowledge, it hasn't been replicated yet. And it's difficult to say whether this will be, a useful technique. And I think in certain neurological disorders, it may be actually detrimental, because in some instances you already have a weakened blood-brain barrier. So, for example, Alzheimer's disease. And I don't know what the situation is in Angelman syndrome yet. I don't think anyone knows. So to disrupt it may be more detrimental because you're already damaging something that's damaged. So, and how that would affect the efficiency of the virus uptake, I don't know. Because a lot of the viruses, they use protein receptors in order to actually move through. They attach to something and then they move into the cell, or they move between the cells. And so it's difficult to say if that would actually help with that method of movement. And it may be great for maybe small molecules or things like that, which have problem getting across the blood-brain barrier, but maybe not so much the virus.
But I mean, maybe an important lesson to take from that study is not even whether it works or it doesn't work, but again, it's just people trying different kinds of approaches, to try to address this problem, because it is a significant barrier for trying to get therapeutics into the brain. There are other types of delivery methods too, some of which Dr. Nash talked about in his talk, that are more or less effective. There's intrathecal kind of delivery as well, intracranial kinds of injection. But of course, people would like to be able to get things across the blood-brain barrier easily, and sometimes that's very hard. So, I mean, maybe this approach is something that may work. Maybe it'll be replicated. Maybe this is not the one that's going to work. But people are trying to make this kind of work. So, I'm encouraged just that people are still pushing on in this area, and if this guy doesn't get it, someone else may be.
And how did yours get across, or were you surprised, or no, did I misinterpret that?
No, I was surprised. I'm always surprised when this stuff works. But I'm happy. I mean, yeah, it was our intention to get it across the blood-brain barrier, but I'm still very happy that it worked.
So since we spent a lot of time in this area, my colleague Mike Kaplan has been working on ultrasound delivery and is a practicing neurosurgeon at Cornell. The problem that you're faced with is that the blood-brain barrier, as we've heard, is there for a very good reason. The moment you open it up, and it's been done for years with gliomas, because we use hyperosmolar mannitol, for example, to open up the blood-brain barrier, get chemotherapeutic drugs ahead. The problem is you immediately allow all sorts of foreign proteins to get in, get a lot of inflammation, get a lot of problems with that. So even though we think about invasive neurosurgery being risky in the context of how many strokes you have and how many complications you have by opening up the blood-brain barrier, stereotactic neurosurgery is far safer as a general rule. The other problem that you're faced with when you give peripheral injection of large amounts of virus, which you really need to do periphery, you think it's less invasive, but to the immune system, it's highly invasive, and so you get some massive immunological response. And remember, only a small fraction of what you're delivering gets across the blood barrier. Most of it's seeded out to the rest of the body. It's a problem. So I think the strategy, and we've thought about this a long time, so there's very safe ways now of injecting things into the CSF, into the brain, using stereotactic process. So I think the energy, even though it's interesting that people are doing it, and I think for gliomas it's promising, I think the future in terms of CNS gene therapy is still going to be standard neurosurgical approaches, which are relatively safe.
And it's possible, I mean, we do this clinically now for the neurosurgeons to place a reservoir that then you just tap into. And yes, it seems invasive, and I know it's a hard decision to make, especially for a young child, for a parent to make. But it's possible you could place a reservoir and then tap into that to deliver